Hello, Namaskar, Khambagani people. Welcome back to our channel, Professor Tiwari, and my name is Amrita. So in the last class, we discussed about the endomembrane system. We just did the introduction part of it from the chapter, the cell, the unit of life from class 11th. So proceeding in this chapter on the same topic, endomembrane structure. So today we're gonna discuss about the endomembrane structures or endomembrane components of the cell. So let's begin with the topic. Endomembrane structures. Okay, now endomembrane structures are those structures which are part of a particular system present in the cell or which has a membrane bound organelles which work with a coordination. Okay, so according to the endomembrane structure, there are 4 types of organelles of the cell which are enclosed or which are part of this endomembrane structures, which is the first one is endoplasmic reticulum, second Golgi bodies, Golgi apparatus or Golgi complex, you want to say, you can name it any one of them. Then next is your lysosomes, and final one is your vacuoles. Okay, so these are the 4 organelles of the cell which are part of the endomembrane structure. Other than this, there are other organelles which are also been there, but they are not the part of endomembrane structure, which means they do not work in a closed you can say in a closed environment or in the close contact with each other. All these 4 which I have discussed just now, endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles, they all work in coordination. Okay, but other than that, mitochondria, peroxisomes and your chloroplast from the cells. These are not the part of the endomembrane structures, which means they work individually. We are not discussing them right now, but we are discussing the endomembrane structures over here. So the first endomembrane structure that we are going to study is about the endoplasmic reticulum. Okay, like I have already discussed the endoplasmic reticulum in the class 9th one. If you want more information on that, the basic information, you can get it from there. But we'll move ahead in the endoplasmic reticulum. Now, if I just make a diagram of an endoplasmic reticulum, then if I just draw it, I'm just going to draw so I'm just drawing the nucleus. So this is your nucleus. Okay, just write nucleus into it. So this is your nucleus. Okay, this nucleus has a nucleopore, which means the membrane has the pores. Alright, just outside this membrane you have this structure. This structure is being called as your endoplasmic reticulum, and you will find many out there just like this. Okay, alright, now moving ahead into this one, first we'll discuss like what is endoplasmic reticulum. So let's just begin with the topic. Endoplasmic reticulum. Okay, this is your ER. This endoplasmic reticulum is also being termed as ER. Okay, in short form. Now moving ahead into the endoplasmic reticulum, see, the endoplasmic reticulum was actually not been discovered until and unless an electron microscope came into existence. Okay, because this endoplasmic reticulum or the structures of inside the cell, they were not visible clearly or easily without the help of an advanced microscope, and that could only take place when the electron microscope came into existence. So when the electron microscope came into existence, it's actually found a network or reticulum of tiny tubules. Okay, so what the electron microscope have seen, like it had seen inside the nucleus, that it has seen a network or a reticulum of a tiny tubule. So there's a tubule-like structure has been there of the endoplasmic reticulum, and they are actually being placed just adjacent to nucleus, which means just outside the nucleus, just after the nucleus you will find the endoplasmic reticulum placed over here. Okay, now moving ahead into this, and the main thing of the endoplasmic reticulum that we are talking about, and this has been called as this whole thing is being called as and been termed as called as endoplasmic reticulum. Okay, now moving ahead into this one, see the endoplasmic reticulum is actually being divided into two parts. So ER divides into two types. Okay, I just tell you how. See, so we have this, if you just see, I have drawn these green things present onto the endoplasmic reticulum. I have drawn it onto one and I have left the other two like that only. So these green ones are being called as ribosomes. Okay. So on the basis of presence or absence of ribosomes, the endoplasmic reticulum are being called as rough endoplasmic reticulum, okay, and smooth endoplasmic reticulum. Alright. Now, the rough endoplasmic reticulum has ribosomes on it and no ribosomes. On smooth endoplasmic reticulum, no ribosomes. Okay. So, on the basis of presence or absence of ribosomes, we classify the endoplasmic reticulum as the rough endoplasmic reticulum, or you can also call it as RER, rough endoplasmic reticulum. And if the ribosomes are absent, which means there is no ribosomes present on it, like this one, it will be called as smooth endoplasmic reticulum with no ribosomes on it, called as SER. Okay. Now, both of these endoplasmic reticulums are different, which means it has ribosomes present onto it and it does not have ribosomes present into it, which means is there any other thing which is been there. Okay. Now, moving ahead into this one, like, does these absence or presence of ribosomes play any crucial role onto it? Okay. Now, moving ahead into that one. Before that, we will discuss one more thing over here. Like, on the basis of endoplasmic reticulum, like on the basis of ER, we actually divide cell into two compartments. What are these two compartments? Luminal, which means inside ER. Okay, and second one is extraluminal. Luminal cytoplasm, which is compartment, which means one part is inside the ER, which is present like this one, and second one is outside one, which is the rest of the cytoplasm that we have of the cell. Okay, so we have the inside of this endoplasmic reticulum is being called as luminal, and the outside part of the ER is being called as extraluminal, which means the cytoplasm of the cell. Okay, now moving ahead, we'll discuss more about the RER, the presence of RER and SER. And the SER, like, does RER and SER have any distinct role, or they play a simple role, or it's just like that? See, you will always find ribosomes present on the endoplasmic reticulum only in active cells. Okay, so if I would say ribosomes present on active cells. Active cells, when I say active cells, that which means that which synthesize proteins. Okay, which means active cells are those ones which are synthesizing proteins continuously, which means the cellular activity has been taking place. So ribosomes will only be present on an active cell, which means the protein formation has been taking place. Okay, so that is the main role of the ribosome. And what does a ribosome do? ribosome synthesize proteins. Okay, this is a sign for the protein. Now, the ribosome alone cannot synthesize protein, which means it needs some companion or it needs some another component of the cell or structure of the cell or organelle of the cell to perform or to form the protein. Now, how it does that? See, what happens is that these ribosomes, they will go and get attached on the endoplasmic reticulum. The mRNA will come from the nucleus, okay, after the transcription, and it will ribosome will get attached onto it and it will take it to the rough endoplasmic reticulum. Okay, we will not go over there because it's a detailed study of that and it's not there in the syllabus too. Okay, and it's part of your syllabus, so will not go over there. Just understand that ribosome takes part in the formation of the proteins with the endoplasmic reticulum. Okay, now what happens is protein synthesis. Protein synthesized by ribosomes. Okay, how it in association with endoplasmic reticulum. Okay, inside the lumen, which means the formation of the protein or the synthesis or the production of the protein takes place inside the lumen of the endoplasmic reticulum when the ribosomes attach onto them. Okay, inside the lumen, and in that one it takes place how it takes place in co-translation process, co-translocation process. Okay. Alright. Now, this co-translocation process is actually nothing but the modification of the protein before it reaches to its final destination. Okay. How it takes place? See, before the protein reaches to final destination, the protein has to undergo 5 modifications. Okay. Now, what are these 5 modifications are? First one, addition and processing of carbohydrates, carbs. Okay. Second is formation of disulfide bonds. Okay. Third one is protein assembly. Okay. Next is your specific proteolytic cleavage. Okay, which means that there are certain sites which has to be present on the protein, so that if required the protein has to be cut and it again it gets turned into amino acids. It takes place during the digestion process of the protein. Okay, when the protein get digested or they have to be degraded. So at that time, if this proteolytic cleavage site has not been there, the degradation of the protein becomes little bit harsh, though the enzymes act on it, but the enzymes should also have a site to act on it. Okay, and the last one is the assembly of multimeric proteins. Okay, which means lot many proteins, like sometimes it happens that many proteins they come together and they make a big protein, okay, a different protein. So these are the 5 processes or 5 modifications that the protein has to undergo inside the lumen of the ER, and that is to the co-translation process, from one process to another process or the transportation of that one. Okay, now this is about this is the function of the RER. But what about the SER, the smooth endoplasmic reticulum? Does a smooth endoplasmic reticulum does not perform any kind of a role, or it's just like it waits unless another ribosomes comes up and attach on it? Actually, it's not the case. The smooth endoplasmic reticulum, if I talk about, smooth endoplasmic reticulum. So smooth endoplasmic reticulum are actually responsible for the synthesis of lipids. Okay, so basically in animals, if you just say animals, what they do in animal cells, I would say, not in animals, I would say animal cells, what they do, they form lipid like steroid hormone, they form. Okay, all right, they are involved in the process in the formation of steroid like hormone and its secretion also. Other than that, they are also responsible for detoxification. Okay, and calcium regulation. Okay, so smooth endoplasmic reticulum performs these functions, which means it is basically involved in the formation of the lipids. Okay, in animal cells, they actually what they form a steroid like they form lipid like steroid hormone. Other than that, they are also involved in the detoxification of the cells and also in the calcium regulation, which means how much calcium has to enter into the cell for certain functions, the calcium ions basically, and how much has to go out. Okay, that is basically a complete different thing that we study under the cell biology, which has not been the part of over here. So this is all about. Now, as we have started, so this is all about the endoplasmic reticulum, and now we'll move to the next one, that is the Golgi apparatus or the Golgi bodies or the Golgi complex. Okay, so next one is your Golgi apparatus. Now, what about Golgi apparatus? Actually, Golgi was being discovered by a scientist called as Camillo Golgi. Camillo Golgi. He discovered Golgi in the year 1898. Okay, how did it found Golgi? So Golgi was actually what he saw a densely you can say densely tuberous kind of a structure, densely sac structure. Okay, he found a densely stacked structure which is near to the nucleus, but it is actually in the close proximation with the endoplasmic reticulum. I just rubbed this ribosome from here just to make the Golgi over here. So I just make a Golgi. Okay, and this is how the Golgi has been there. Okay, and I'll just make two things over here more. These are not the ribosomes, we'll discuss what they are. Okay, like this. Now, this is our Golgi. Okay, this red one is a Golgi. Now, he found a tubular like structure, and it actually what he saw is he saw flat, or you can say disc shaped or cistern. Okay, so Golgi's these ones, this structure of Golgi has also been called as cistern. Okay, now this was the total structure of the Golgi. If we just talk about now the structure of the diameter, if we talk about the diameter of the Golgi, the diameter of the Golgi is actually between 0.5 to 0.5 to 1 micrometer. So this is the diameter of the Golgi, basically you can say it, and the number of Golgis, they also vary according to the cell. Okay, now what does this Golgi actually do? If you just look at the structure of the Golgi over here, you will see this one and this one is quite different from these ones. Okay, why is it so? This one is being called as the convex cis phase. Okay, so this one, so Golgi has two phase. Okay, first one is, I would say, the cis convex, distinct convex cis phase, distinct convex cis phase, which is also been called as forming phase. Okay, and the second one is being called as your concave phase, concave trans, or it is a concave trans phase or mature phase. Okay, this is cis convex phase, this is trans concave phase. Cis convex phase is always been near to the nucleus or near to the endoplasmic reticulum. Why is it so? We'll discuss. And this is always away from the nucleus, which is making a concave shape, which is outside. Okay, forming this form. Now, if you just look at the structure, then this convex phase and this concave phase, they are actually different from each other, but still they are interconnected. Okay, they are different. This cis and trans. If you just talk about cis and trans, different from each other. Okay, but still they are interconnected. Okay, now moving ahead into this one, see what happens is, what is the role of the Golgi over here? Like we have seen, like, okay, it has two faces, it has this one, this is the lumen of the Golgi. What they are being involved? They are actually being connected with the rough endoplasmic reticulum or with the smooth endoplasmic reticulum for the transportation or for the modification of the proteins. Whatever the proteins that has been formed in the lumen or in the endoplasmic reticulum, they are actually being transported to the Golgi in the vesicles. So these ones, which green ones, which I told you, which I'll tell you later on, these are the vesicles, and these are also vesicles. Okay, now what happens is, so what they do, these vesicles which are coming, they actually come from the endoplasmic reticulum carrying the protein. Okay, they are coming from the endoplasmic reticulum carrying the protein or carrying the lipids, you can say. So what they do, vesicles originating from Golgi, from ER, what they do, they actually fuse into Golgi. Okay, when they fuse into Golgi, inside the lumen of the Golgi, what happens? There is a modification of proteins and lipids takes place. Okay, the modification of proteins and lipids, more of the modifications takes place. All right, and once it has been done, so they are being transported like from here to here, then vesicle will fuse from here to here, another vesicle will form and will fuse from here to here, and lately it will be come out like this. Okay, so this vesicle which is over here and this vesicle over here is completely different. They both are carrying the protein, but this vesicle will go and fuse into this cis phase, then another vesicle will form and will get fused into another one, and another lumen of the Golgi, another stack of the Golgi, then in the trans phase, and then it will come out with the modified protein, and that protein or lipid, I would say, what it will happen with it will be dispatched to intracellular, either intracellular site where it has been required, okay, or into the cell somewhere else. Okay, might be in the intracellular site somewhere in the cell or somewhere else, wherever the protein is being required. Now moving ahead into this one, see, as I told you that Golgi is involved in the modification or in the formation of proteins, and this one, so Golgi is also the site of, if I would say, Golgi is also the site of the formation of glycolipids and glycoproteins. Okay, so the proteins that has been modified or the lipids that has been modified will be called as, so site, so we can call it Golgi is the site of what? glycoproteins and glycolipids. Alright, so this is all about the Golgi apparatus. Okay, now we'll move ahead into the third one, that is the lysosomes. So we'll quickly look at the lysosome of the cell. Okay, I just rub this diagram out. If I just make a diagram of the lysosome, okay, this is the inside portion of the lysosome. Okay, another layer which comes up over here. There's one more layer over here comes. Okay, and one more layer over here. I'm just making this outside layer as a double one. Okay, and I'll tell you later on why is it so. Okay, all right. Now this one contains hydrolytic enzymes mixture. Okay, inside, just inside. Okay, next is this green one is actually the lipid layer. Okay, and in this one, the outside one, these one are glycosylated molecules. Okay, now talking about lysosome. So this is the structure of lysosome that we have. Now if you just look at the structure of the lysosomes are actually the single membrane structure. Okay, they are the single membrane structure. If I talk about single membrane structure, okay, now if you just look at this one, it is being made up of lipid layer. Okay, next, this lysosome is actually the site for the degradation or for the basically by the degradation or for the removal of the used carbohydrates, lipids, nucleic acids, different biomolecules which are present inside the cell and which are no longer useful, or which has already been used and they are not being used more. So they actually been removed with the help of lysosomes. So lysosomes actually have acidic pH. This acidic pH is actually around 5. Okay, and what it contains in this acidic pH, it contains the hydrolytic enzymes. These hydrolytic enzymes, what are these hydrolytic enzymes are? I just name them over here. These are your hydrolases, okay, lipases, nucleases, proteases, okay, what more? Glycosidases, phospholipases, phosphatase and sulfatase. Okay, so these are the various range of hydrolytic enzymes which are present over here in the center. Okay, that is why the pH is very low. And what it actually do, these enzymes actually they degrade, I would say. They degrade, what they degrade? They degrade carbs, okay, they also contain carbohydrates. So what they degrade? They degrade carbs, lipids, proteins, nucleic acids, okay, any DNA, RNA which has been present over here. They actually, and how these actually been formed, these lysosomes are actually being formed by the Golgi only. Okay, these are the vesicles which are formed from the Golgi, and that is single membrane structure. Okay, they have a lipid layer being formed with the lipid layer, which means the glycoprotein, that is the glycolipid which is being formed, they actually being formed in the form of vesicles, in the form of lysosomes, and they have the hydrolytic pH just like the lower one, around 5, and they have the similar, they have these different type of hydrolytic enzymes, hydrolases, lipases, nucleases, proteases, glycosidases, phospholipases, phosphatase, sulfatase, and carbohydrates. And what they degrade, they actually degrade all the unwanted material of the cell which is already being used or which will no longer be in use in the future, or just the waste material of the cell, which degrades the carbs, the lipids, the proteins, the nucleic acids. Okay, not the waste material, but these biomolecules which has already been used and will be no longer in the use. Okay. Okay. All right. Now we are being left with the one more endomembrane structure, that is your vacuoles, and that we'll do it in the next class. I hope you don't have any kind of a doubt, and if you have, you can drop the question in the comment box. I'll answer that one. And if please like, share, and subscribe our channel, and don't forget to hit the bell icon for more such updates. Thank you. Bye.